See my screen. I don't see you, so if you can just tell me if it's
Looks
okay.
good.
Looks good.
Thank you. Okay, great. So, welcome to this webinar series that is hosted from the SCOR working group for the BGC about the coordination of the development of gridded four-dimensional data products from BGC Argo observations. So this working group aims to facilitate the discussion and the coordination among different scientific communities around the development, the validation, and the distribution of the 4D BGC products from observational datasets with a focus more on the BGC Argo array. So the goal is to significantly enhance access and utility of BGC Argo observations through the 4D BGC products. So you have here a picture of the group that has met during the OSL meeting in Glasgow last month. So here I wanted to say that we have a web page on the BGC Argo website where you can find all the 4D BGC products. So if you are interested in these products, you can go to this web page to see all the products based on BGC Argo observations. The working group has also a website. So if you are interested in having the news and see the activities of the group and so on, you can go on this website. You will have all the information about the group. You have also a mailing list to receive all the updates on the 4DBGC working group activities and the webinar series, for example. So you can sign up to receive the updates. All the webinars are recorded and you can find them on the YouTube channel of the group, the 4DBGC working group channel on YouTube. And today we are very pleased to have Nathan Briggs that will give us a talk on the creation of a global climatology of sinking organic carbon flux and mean particle diameter from Argo optical backscattering data. So thank you, Nathan. And I will stop sharing my screen and you can share yours.
Let's see. Do you see my
Thank you.
screen?
Yeah, perfect.
Hi, everybody. Yeah, I'm quite excited to talk about a product that we've been working on for quite a few years, and it's hopefully ready to distribute later this month. So, yeah, I think we can add a link to it on the 4D BGC web page as soon as it's live. It's not quite there yet. So what is this new data product from BGC Argo Floats? This is a sort of open ocean global climatology of particle size and sinking particulate organic carbon flux, all from optical backscattering measurements. So I want to make sure I thank my co-authors who have helped me with this research and also the funding sources below. So what is the source of this data? This is the Biogeochemical Argo network of floats equipped with optical backscattering sensors, and it's using all data between sort of the first floats deployed in 2010 and through July 2025. So here's a map showing as of 2020, July 2025, what were the sort of live floats and previous floats? So we have 1,300 floats in total contributing to this data set. And so briefly, for those who aren't familiar with BGC Argo, these are floats that profile every 10 days at least between 2,000 meters and the surface. And Argo floats carry temperature and salinity sensors. BGC Argo sensors floats carry other biogeochemical sensors, of one of which is optical backscattering. Why do we care about these products of particle size and flux. The reason we care about this is that we think that large, fast-sinking particles drive a lot of the biological carbon pump that stores carbon in the deep ocean, keeps it away from the atmosphere, and also feeds deep ecosystems. Uh, so it's quite difficult to measure sinking particles that get below the surface to a depth where carbon can be sequestered long time series, long for long time periods. It's quite difficult to measure these at scale, and so BGC Argo has offered one of the first opportunities to have a really global look at these sinking particles across all seasons. So how are we? I'm saying that optical backscattering is a biogeochemical sensor, but those who aren't really familiar with this sensor, it's not quite true. It's really an optical sensor looking at particles in the water, whether these particles have any biological content or not. But we can use it for biogeochemistry in the open ocean because a lot of particles are organic. So what's the principle of the sensor? You have a light source, you shine light into the water. The more particles are in the water, the more of that light gets scattered back to your detector. And so on the left, you have high particle concentration, on the right, you have low, and you have less light on the right getting back to your detector. And that's really all the sensor does. Um, so how do we get from this measurement to particle size? And this is taking advantage of a method that takes advantage of the very small sample volume of these sensors. So on the right, you can see one of these backscattering sensors shining its light into the water. And within the sample volume, you have a number of small particles. And then up above, you have one big aggregate of smaller particles. So as the water moves past the sensor or the sensor moves through the water, you get repeated measurements of very similar concentrations of small particles. But then when one of these large particles comes through, it's large and rare relative to the sample volume, so it creates a big spike in your backscattering measurement. And then when it passes out of the sampling volume, it returns to the sort of background, more continuous background caused by many smaller particles. So you can think of these as sort of individual cells of plankton or bacteria, very small detrital particles in the water. And you can think of the large particles, big aggregates or fecal pellets that might sink fast in the water. So once we've captured these data, including these spikes, we can separate the spikes from the large particles from the sort of background, more continuous background from the smaller particles, so that we can create this sort of digital filter in our time series or a profile of backscattering measurements. And we can separate our small particle backscattering from our large particle backscattering. So here's an example of a profile of particle backscattering in the ocean. Ooh, my labels say POC, but this is really backscattering, and we'll get to the carbon conversion later. Um, so using a digital filter, we get the background of small particles in orange when there's no large particle spikes coming through. And then you get your spikes from your large particles are the difference between your blue line and your orange line. We can go further than this sort of large versus small dichotomy, and we can relate the diameter of the particle that creates a spike, as long as we can assume that each particle creates one spike. We can relate the diameter of the particle that could have created that spike to the backscattering the height of that spike in units of backscattering, the sample volume of the backscattering sensor, the backscattering efficiency of the particle. And so these sample volume is known, and the backscattering efficiency of particles has been empirically constrained on average in the ocean. So we're going to use an average value to get a first indication of the particle diameter. So these are some examples of different particle heights corresponding to different size particles. And so the simplest way to look at this data is simply to add up spikes of different height, convert that into into particle diameter and divide it by the total amount of volume that your sensor has sampled. And then you can, over a given depth bin, you can get a distribution of particle concentrations of different sizes. Before we do that, we do have to do quite a lot of QC of the data. So this is some raw time series of both the small particle signal on the left, which is the smooth, continuous filtered signal, and then the large particle signal on the right, which are these individual spikes. And we can see this sort of banding pattern in both the small and the large particle signal in some places that's often indicative of fouling. So sometimes we get a apparent fouling of the sensors that creates just spikes without much in the background. And sometimes we see elevated background without spikes, and sometimes we see a bit of both. So we need a quality control method that gets rid of potential fouling in both the spikiness and the background. So here are some examples. And then here, the lower two rows are an automated QC procedure that we went through to take a first pass at removing anomalies in so single profile anomalies in both spikes, large particles, and background, small particles. And we can see here that it removes a lot of the obvious signals of fouling. So, but it leaves some of this more gradual fouling that you can see in this black box here in our small particle signal. So then we have another visual QC procedure that we go through. We look at every float time series in this way, and we pick the first profile that we think um, fouling started at, and we remove this. This is somewhat duplicating some of the QC that's going on in the Argo data system, but we've sort of developed this independently for now. Um, okay, once we've done this QC and we're convinced that our most of the spikes that are left are due to actual particle encounter in the ocean rather than fouling, we can then validate these particle concentrations at different sizes using floats that are equipped with particle cameras. So this is a map of 27 BGC Argo floats that have been deployed with particle cameras that can much more accurately measure particle size. Um, Most of these were deployed by the Refine project, although there were a few other contributions, and they're all part of the BGC Argo data system at the moment. So with a lot of binning, well, I've been averaging, each point in this validation data set is a 15 profile average, and we need to do a lot of averaging to get good statistics on these rare spikes. But when we do this, we do find pretty good agreement between basically counting up the spikes of a given height and converting that to a size, and the UVP camera-based um, particle counts. Um, so on both the X and the Y axis, we have particle concentration of a specific size class per liter. So how many particles per liter? And we can see on the left, we have 250 to 300 micron particles. On the right, we have 800 to 1,000 micron particles. And within this range, we're getting fairly good agreement between the backscattering derived concentrations and the UVP6 derived concentrations. A slight overestimate using the backscattering technique. Mean bias between 16 and 46% and R squared in this range, always over 70%. When we get, oops, sorry, this label is wrong. This is 200 to 250 microns. When we get to the smallest particles very close to the surface, they don't come through the sample volume one at a time. So we start to underestimate. And so you see the UVP when you get very close to the surface, the UVP concentration on the right goes up to 100 particles per liter. And the backscattering cannot measure that many particles because it would lose sight of the small particle background. And so these pink X's are where we've identified a priori that there's too many particles to accurately get the concentration in this size class. Um, so it's one of the limits of the method. When you get very close to the surface, for the smaller particles, they're so numerous that you kind of lose track of what's the background and what's the individual spikes from individual particles. But within this size range, 250 to 1,000, we're doing pretty well. So here's the results of now applying this to the global climatology. So what I've done is taken all the profiles from all the floats in the entire time period, 2010 to 2025, and taken the concentrations of particles. In this case, I'm looking at particles between 200 and 400 microns, and I'm looking at depth between 150 and 300 microns. So we're getting away from the surface where we can sometimes get this undercounting in this particle size class. Um, so yeah, I mean, this is the sort of first look we've had really in all seasons across all of the oceans of particles of of a different of particles of a certain size class. So these are concentrations again per liter of 200 to 400 micron particles, 150 to 300 meters depth. You can see that the signal is a bit noisy. In certain pixels, we have more or less data, but we can very clearly see the sort of seasonal patterns in appearance of these particles at depth. We can do this for different size classes. So again, we're looking at the 150 to 300 meter um, depth range. And here I'm zooming in um, on just a single month, just January climatology. And we can look at the backscattering background. So that's particles smaller than 200 microns on the upper left, the 200 to 400 micron particles, 400, 800, and above 800 micron particles. So you can see that um, the upper left is in units of backscattering, but the others are units of concentration per liter. And we can see the concentration go down. And you can also see the sort of latitudinal gradient get sharper as you go up in size. We can also look at any of these given concentrations as a function of depth. Um, the scale bars are, color bars are changing with depth, but this is looking at 400 to 800 micron particles um, near the surface, and then in the upper mesopelagic and the lower mesopelagic. And you can see the attenuation with depth. You can also see as you get deeper, the signal does become a bit noisier, where a single particle can cause local anomalies, but we can still clearly see the sort of regional patterns and the large flux to the deep ocean in the Southern Ocean in January. Related to this concentrations of different sizes, we can also look at the data a different way and get a sort of average size and see when the sort of ratio of large to small particles changes throughout the year. And again, we can see at the high latitudes in the productive seasons, not only are there more large particles, but the average particle size is bigger. Um, so this is, again, the surface, near surface climatology of mean particle size throughout the year. Again, the product is a bit noisy because it's relying on these rare encounters with spikes, um, but the seasonal patterns and the basin-wide patterns are pretty clear. So that was size. We also want to, one reason we're interested in these big particles is how much carbon they're bringing with them to the deep ocean. So how can we estimate POC fluxes from this size fraction in backscattering? There's a couple steps involved. The first is to convert uh, optical backscattering into units of carbon. So for this, we use empirical relationships On the left, we see an example. The data points are one study, Johnson et al. 2017, which is Southern Ocean, and it's looking at near surface relationships between POC and optical backscattering. And then the different colored lines are different studies that have found different relationships in different regions. So that's the near surface relationship. And so we use a single sort of near surface relationship as our first guess, and then we take a range from the literature as our uncertainty. And then there's also a depth dependence to this conversion. So as particles sink, particles that originate in the surface, as they sink and get reworked, they lose their organic fraction faster than they lose their total mass. So we need to adjust our POC to backscattering ratio with depth. This solid red line on the right is showing the mean um, depth attenuation that we impose on this data set based on a couple different data sources. One of them is this Septinich et al. 2012 data set from the North Atlantic, um, showing depth-dependent POC to backscattering ratio. And the other source of data we use is sediment trap data, global sediment trap data set, looking at the ratio of organic to inorganic matter with depth. These different dashed and solid lines around the thick red line are different sources of uncertainty in this ratio, which we're going to propagate into any POC flux estimates that come from these data. Again, we're going to use our spike method to partition. Once we convert backscattering to POC, we partition that POC into three size classes based on the background, small spikes, and large spikes. And then we assign each of those fractions its own sinking speed based on literature values with their own uncertainties. So the small sinking, we assume the small particles we assume sink at 2.5 meters per day, the largest 74 meters per day. Um, so when you multiply the POC concentration in the different size class by its sinking speed, you get the flux, downward flux, and then you add up all the different size classes and their own fluxes, and you get the total POC flux estimate from this method. Because this includes, even though the sinking speed is quite small, sometimes the small POC dominates the flux, especially in deep values. So it's really critical to get this deep value right. And there's a lot of reasons that you can get an offset in your deep backscattering value. Um, so uh, in order to address these issues before calculating this global flux climatology, we first want to align different sensors at depth because there can be calibration differences between backscattering sensors on different floats. So this is showing I think the steps that we took to align sensors at depth before we make our global small particle climatology that forms part of our flux estimate. So the first step is to find matchups between any two floats that are within 400 kilometers of each other in the same month of the year. And then for each float, find a median deep offset relative to all other floats, and then subtract this offset. So it basically just aligns floats with floats it's encountered in its lifetime in the same month of the year. Um, so on the right, we see a time series in red of backscattering at 400 meters from a particular float that took this trajectory. And then the colors around it show other floats nearby that it encountered. And you can see this one float that it encountered had a much higher backscattering. So this is very likely a calibration issue for that one float. So the next slide shows the same time series before and after alignment. And you can see that it hasn't brought them into perfect agreement, but it's brought them into much closer alignment and removed this outlier. So I think this helps with consistency of this method. And then the other step we take after we've aligned nearby floats Okay, I guess this can show you the result of that. So the result of this alignment exercise, we're looking at our POC due to small particles climatology in the lower mesopelagic before and after this alignment exercise, and you can see much smoother spatial patterns after the alignment. Um, and then we also often see this sort of deep, constant background of backscattering in the ocean, which um, is we think is probably not due to actively sinking labile particles. Um, and so the next step before calculating the carbon flux is to subtract this background. And the way we do this is we find um, for our climatology, we find the smallest backscattering at any depth at any time of the year for a given grid cell and subtract that. So this refractory backscattering correction includes both regional variability in calibration and also regional variability in potentially refractory particle pool. And that could be mineral particles, that could be very fine refractory POC as well, that we don't think is actively involved in the sort of annual pulses of sinking material. Okay, so now we have a final POC flux calculation. We've subtracted this refractory background so that our small labile POC that we think might be contributing to the flux, that's the difference between our smooth of small particle filter and the refractory background. And then the difference between the sort of orange and purple is our medium particles. The difference between purple and green is our large particles. And then we can add up each of these contributions to flux, and we get our final flux value from each flow that we then grid again, 4 by 8 degree grid, um, 50, 50 meter vertical bins, and monthly climatologies. So this is showing monthly POC flux at 200 meters. It looks, similar to the particle concentrations that I showed you, but this is including all the contributions of all the particle sizes altogether. And this is now in units of POC flux, milligrams per meter squared per day. The final step to make a truly sort of global climatology is an additional smoothing and interpolation step to fill in all the gaps. Uh, and then we can validate this against other measurements of POC flux in the ocean. This is the validation regression versus the Mao et al. 2016 global compilation of sediment trap and thorium disequilibrium based sinking POC flux estimates. So you can see that with the parameters that we've chosen, the sort of mean parameters we've chosen to convert to POC and to flux, we're getting fairly good agreement with this global data set across a wide range of depths. So color is depth. Circle size in this plot is how much backscattering data is in each of these. And these matchups are climatological matchups. So any sediment trap data or any thorium-based data estimating downward POC flux is matched against its nearest climatological grid cell in that month of the year. So the scatter around the one-to-one line includes methodological errors in this flux estimate, but it also includes spatial and temporal differences. So first order, we have a relatively low bias across a wide range of depths and a wide range of ocean conditions, but there's still quite a lot of scatter around this line. This is sort of zooming in on individual time series, long-term time series of sinking carbon flux, specifically the hot time series on the left and the bats time series on the right. And these are places where sediment traps are deployed monthly over many years. So we get good climatology in these regions of what the annual cycle in carbon flux should be at multiple depths. And so this is showing 150 meters and 300 meters. Um, and the purple one is this globe sync data set that I've been telling you about. And then the red and the orange and the yellow lines are the mean and the median of the sediment trap data. Along with the globe sync estimates, we have estimates of systematic uncertainties from propagating uncertainties in sinking speed and POC conversions. We also have estimates of precision, and um, that's due to uncertainty that comes from rare encounters with these large particles that create these spikes. So the fewer measurements that we have, the wider these precision bounds are. So you can see at hot we're agreeing with the sediment traps within the sort of stated uncertainties in basically every month of the year. Uh, Although there's a sort of a small systematic difference where GlobeSync is slightly overestimating. At BATS, we can see the GlobeSync is sort of right on most of the year, but there's events in the first 2 months of the year where GlobeSync is underestimating. And I'm just going to zoom in on this discrepancy in January and February, just as a good example of how, this data product is really nice because it's global, it's consistent in its method, but it still seems to be missing some things in certain moments. So it looks like in January and February and March at BATS, um, we have quite low stated uncertainty in this globe sync product, but we're dramatically underestimating the measured flux. Um, so it seems to me most likely that these are sinking particles that either are too big or sink too fast for them to be picked up reliably in this backscattering based method. So just a word of caution, when you're interpreting the product, that in specific times and places we might be missing some sort of extreme events. Okay, with that caveat out of the way, what is the sort of global mean sinking POC flux profile that comes out of this product? So this is sort of the global amount of carbon stored per year by sinking flux, at least the subset of sinking particles that can be seen by these backscattering measurements. And we get an annual number of almost 8 petagrams per year sinking below 100 meters and zero point 0.5 petagrams per year sinking below 1,000 meters. We can also compare this to other estimates of other sort of broad scale estimates of sinking POC. These are exports, so export out of the top 100 meters by various models that rely on satellites. Just to highlight a few, there's some food web based methods that don't correlate very well with the GlobeSync product. There are some empirical methods that have looked at empirical relationships between satellite net primary productivity products and independent uh, estimates of export. And they perform a little bit better, especially the ones based on the CAFE NPP and primary productivity model. Um, Plus some empirical correlations with export efficiency. We're getting our highest correlations. Oops, I didn't mean to. I didn't mean to include those. Well, I guess it's worth saying that these data come alongside of um, other average. I've averaged other parameters from this data set on the same grid, just for convenience, that come from the floats, including chlorophyll, including the POC at the surface. Um, so we can now use these data to examine what controls particle export at global scale. We can look at the relationship with temperature. Um, we can look at the relationship with surface chlorophyll. Um, Not a great correlation. We can look at the relationship between deep export of carbon and surface particulate organic carbon concentration. That looks a little bit better. We can look at the relationship between the surface size parameter and our flux, our export flux, and that looks a little bit better. If we combine surface concentration and surface size, we get an even better predictor of our deep carbon export. And what about sequestration, going deeper? So on the left, again, I'm showing you surface POC concentration times surface mean size on the x-axis, and the y-axis 200-meter export, and on the left, and 700-meter export on the right. So that's a sort of sequestration flux, the deeper storage. And even though the R squared goes down, it's still a pretty good predictor. The surface size and surface concentration of particles in this data set are still a reasonable predictor of a lot of the variability by in spatial and seasonal variability in our deep carbon flux. So the submission of this data set is now in process. Hopefully, it should be available for everybody very soon. It's a global scale surface to 2,000-meter monthly gridded climatology of POC flux and particle size and concentration. Um, the validation shows very little mean bias. So for large scale analyses and comparisons, I think you you can be confident it won't have a large bias versus sediment traps at global scale, but just warning that it can deviate substantially at local scales where specific processes it may not capture specific extreme processes. Um, and then it comes alongside key ancillary data from the same BGC Argo floats. So the size, chlorophyll, oxygen, nitrate, pH, temperature, and salinity. And yeah, I just want to thank the Global Argo program for making this all possible. It's really incredible to have global coverage that you can apply a single method to get comparable quantities across basically the full open ocean, excluding the Arctic. So thanks. Yeah, I'd be happy to have answer any questions.
Thank you so much, Nathan, for this very nice talk. So now we can take some questions if you have. Yes, Stevie.
Hi, Nathan. Great talk. I was wondering if you have a map of like the climatological transfer efficiency from GlobeSync. I'd just be curious to see what that looks like.
Good question. Let me see if I have it anywhere here. Um, let's see. I let's see. I think I have it in another place. So I did an analysis of transfer efficiency on an older version of this data set, and I've made some changes since then. Um, and basically, long story short, I wasn't able to find any really clear driver of transfer efficiency, but let me see. I can show you what I got. Um, so this was my attempt, and the results may have changed a little bit because I've been tweaking the method per publication. But this is the flux attenuation coefficient and different things that I'm trying to use to predict it. Um, Flux attenuation coefficient is it's not exactly transfer efficiency, but it's higher when you lose more flux with depth in the ocean, and it's lower when you transfer more flux deeper. Um, so the first thing I want to look at is temperature a good predictor? We think if um, if remineralization is driven by temperature, you might expect higher flux attenuation at higher temperatures, and we don't really find that. We actually find a slight opposite trend, but very, very small. We might think that oxygen, low oxygen, you might have um, higher transfer efficiency and therefore lower flux attenuation exponent. And we don't really see that either. Um, uh, the best predictor of flux attenuation is just how much flux. Um, so the more flux you have near the surface, the more attenuation you have. Um, Part of that is could be sort of a statistical artifact that um, the attenuation with depth is sort of the ratio of the shallow flux, I mean the deep flux to the shallow flux, and so that ratio is going to get less if you have a higher value, a shallow value. Um, but there's also, it can be a biological explanation for that, which is when you have more particles, there's more things feeding on those particles in the water. And so you might expect higher flux attenuation when you have a big pulse of export. Um, if we do zoom in on the really high flux events, we can see um, a second order um, pattern coming out. And this is a bit speculative based on really a few data points. The whole trend is based on a few data points. So take this with a grain of salt. But if you look at just the high export events, we can have this proxy of fragmentation, which is the ratio of small to large particles. So you have a bunch of large particles sinking. If they're fragmenting into small particles, they attenuate, they stop sinking, and they don't get as deep. So if you see a lot of small particles showing up during a large particle event, you're going to get this higher ratio of small to large POC, which is our fragmentation proxy. And then we see higher flux attenuations with higher apparent fragmentation. So fragmentation events, when you just isolate very high flux events, might be another driver. Um, Also, when you look at the high flux events, this temperature relationship is now changed from what it was when you include all the data. So it's a sort of complicated mess. I don't have an easy answer for you, um, but we have some interesting indications.
Thank you. We have a question also on the chat, sorry, just before the other question. How can we represent the remineralization?
Um, Good question. So I guess that's two questions. Um, I suppose we could, if you look at the loss of POC with depth and you assume a sinking speed, you can convert that into a loss per day of material. That can include, if you're looking at a specific particle size, that can include consumption by zooplankton. It can include fragmentation into a smaller size class, and it can include remineralization. Um, and But there's also some remineralization, assumed remineralization embedded in the depth-dependent POC to backscattering ratios. So I've already kind of imposed some assumptions about some remineralization happening in the model itself. So then these data can't directly be used to investigate that. I mean, the best way we can look at remineralization with BGC Argo products is probably looking at oxygen and patterns in AOU, seasonal patterns in the upper ocean, or AOU divided by water mass age. And we can try to then relate this attenuation of flux to remineralization. Because basically, if you subtract the flux at one depth from the flux in another depth, you have a net supply of carbon to that layer, and there should be remineralization that balances that out.
Thank you. We have you can.
Yes, can you hear me? Yeah. Hi, Nathan. So great talk. And I have two questions. And the first one is about the filter you mentioned. So how does it filter out the spikes from signals of small diameter particles? And the second question is about the method of your dividing particles into three categories and use it to estimate flux. So what's the logic behind this? Can we do a further subdivision of particles for maybe a better estimation? Yeah, thank you.
Yeah, great question. So the filter, I use a running minimum followed by a running maximum. And it's a bit of a strange thing, but the running minimum is to try to get the background when we have lots of spikes. Um, and then the running maximum after that is to correct for if you take the running minimum on a gradient, you're gonna get the wrong baseline. So it's a running minimum followed by a running maximum. And then there's a further correction for the contribution of noise to the spike signal. Um, and then yeah, that's a great question about how many size classes should we use? Yeah, I think you're probably right. Technically, we could do better by further refining into more size classes. A lot of times when you look at particles bigger than a couple hundred microns, when we look at the size to sinking speed relationship, empirically from measurements of sinking speed, often the relationship is not as good as we would hope. So there may be other drivers of sinking speed other than particle size, like its density becomes important. So I kind of took this originally. I just had large and small, and very recently I divided into three. So to me, this is exciting. Now I have three size classes. But you're right. I think there's no really reason that you couldn't do this further. Just we don't have great information about what sinking speed to assign to these different sizes.
Okay, thank you.
Thank you. Hervé?
Yes, thank you, Nathan. It was very, very interesting. And I have two questions, at least, but I start with two. The first one is with respect to the sampling resolution of over the vertical, according to the diversity of float. What is the impact, and what could be a recommendation for BGC as a whole to have optimize sampling resolution to improve your product?
Yeah, that's a great question. So, yeah, it is a big issue. Really, the best sampling resolution is 1 to 2 meters sampling resolution, because the in a lot of depths and a lot of places, the major limitation on this method is just not enough data points collected. So um, this precision estimate becomes really wide in sometimes in places. And when you see how noisy the climatologies look, most of that is due to just not high enough sampling frequency. So I would really recommend sort of sampling every meter. The caveat with that is that sometimes when you're sampling constantly, you get zooplankton attracted to the sensor that create these extra spikes. I think some of the newer sensors, like the Tridente sensor that is being experimented with, that turns on, it can turn on and turn off very rapidly, and so that sensor might be really even better for this method because you could sample every meter, but it's only on for 1 second and then off for 9 seconds. Then the zooplankton don't follow it, so you can get a high resolution without this zooplankton contamination. But in most times, most depths, um, uh, even existing sensors, 1 meter resolution is really good. And even if possible, all the way down to 2000 meters. I know that it's a lot to ask, but yeah, a lot of places in the ocean, we have data but it's really uncertain because we're only taking one measurement every 10 meters and we're just not finding enough spikes to do good statistics on.
Okay. And the second question is the method that you use to offset it to make the floats from a given area to converge. And this recall me that when BGC Argo is doing delayed mode data, it's float by float, but sometimes there are Argo regional center, the mandate of which is to reharmonize the data from a given area just to have a in fine better view of this area with respect to a parameter. And I was wondering if that maybe is for the BGC Argo data management community, how we could transfer your methods to have data sets that in fine improve each delayed mode file for given floats. So this is for sure something that come after that could generate reprocessing. So this puts some, I would say, pressure on the DAC, but this might be also an option to revisit sometimes when we do reprocessing, and I'm seeking to a reprocessing of chlorophyll A that will come soon, at least in real time, that could help also to have better and better data set.
Yeah, it's a good point because almost all of the BGC sensors need some kind of alignment at depth. So chlorophyll A, you can subtract the deep background.
Yeah, for sure.
Oxygen, nitrate, pH, you all align those to deep climatologies. These sensors all have calibration uncertainties, and backscattering is the only one that we don't have a climatology, so we have no way to align to a known climatology. So the only way we can do it is align
Yeah.
them to each other. And it's only recently that we have enough floats and enough coverage that now most floats have at some point in their lifetime come near another float at the same month. So we do have this new opportunity to align at depth.
Yeah.
I've been talking with Raphael and her SOCA product, looking at deviations from that SOCA product, maybe another way to align them at depth that maybe be easier for
Yeah.
DACs to implement than my method. So I think that might be a good path forward. We have to double check that it's giving good answers and not sort of entrenching biases from old data. But I think in order to harmonize and to take out these big outliers, a float that has a sort of
Yeah.
bad calibration, I think that might be a good first step is aligning to SOCA, basically using SOCA in the way that
Okay.
we use these other climatologies at the
Like
DAC
an
level.
interpolator. Yeah.
So actually, I did it and I have some first results, and it's quite nice because it's aligning with your offset. So I think it will be maybe a way to go for the delayed mode for BBP.
Yeah.
Maybe not in real time, but at least for delayed mode.
And just a last question. So the Mediterranean Sea in your map is very small. That's normal. It's less than 1% of the ocean. But I notice that when you work on the refractory part, the eastern basin seems clearer than the western one. So I was questioning something there because it's a sea that is very close to the Saharan dust deposition. And at some point, I was thinking that this refractory BBP could help to this, but it seems not. So
Yeah, I think the Mediterranean because there's so many floats deployed there, that we
yeah,
could apply a
exactly.
finer grid and do a better job
Yeah, yeah, yeah,
there.
yeah,
So I think a different version of this analysis could be done that would be really interesting. Basically, in
yeah,
any of the regions that have higher coverage, because these really wide, big grid cells were kind of aimed
yeah,
at the
yeah,
regions of the ocean that have low coverage. Um, but yeah, I think there's no reason that we couldn't
yeah, yeah. Good point. Yeah.
do a higher resolution version in the Mediterranean.
Okay, thank you.
Maybe a last one in the chat. So there is a question on how to calculate the median and large POC concentration from a single spike signals.
Sure. So Yeah, I kind of went through that very quickly. So the the medium and the large POC concentrations from the single spike signal. So we convert the backscattering into units of POC. So every spike, um, the height of the spike is now a height in POC concentration. Um, so basically, you add up all the spikes of a given size range. Um, you add up their contributions to POC. So another way of saying it is you take all spikes between a certain range of heights, set everything else to zero, and then you take the average POC across a certain bin. I don't know if that was very clear or not. Um, but basically, your backscattering is in units of POC, and you basically divide your that POC signal into all of the data below a certain spike height. That's your small. All between a one spike height A and spike height B. That's your medium. And it's already in units of POC because you converted your backscatter to POC with your with the empirical conversion.
Okay, thank you. And thank you so much for everything, the talk and the
Yeah,
questions.
yeah, thank you, Raphael. I'll let you know when it's published if you want to link to it. Yeah.
Yeah, and I will have some questions, so I will come back
Okay.
by
Okay,
mail.
great.
Thank you so much.